I would like the discussion to evolve around the implication and how this is going to be changing practice.
But you're going to behave as a cardiologist, and I guess I'll behave as a statistician, Stuart.
Speaking of regulatory issues, and Angelos and Gonzalo, you can.
So I would also like to thank Fayaz for the opportunity to come and talk about this today.
And we published it in circular. Has to do with the time of the measurement and the time of the procedure.
Spontaneous myocardial infarctions, the ones that occur after the patient leaves.
And I think it has great utility. It lets you look at how
This was in America, not in Europe.
We wish to keep this going, this special session on the trials of the year, because it's really very educational for lots of us, at least certainly for me. I would like really to thank all the speakers here and the discussant, the panel. I thank you, all of you, for this wonderful, lovely discussion.
I'm a Duke Cardiology Fellow. I'm honored to have with me Rick Geller, Director of Biostatistics at the NHLBI. Dr. Geller, as a cardiology fellow, cardiology is very rich in clinical trials, and at least when we read about clinical trials, we've seen an evolution from the 70s and 80s to current clinical trials. So in your opinion, how have the statistical methods changed, and where do you see them going in the near future?
So we have lots of new statistical methods. I think many of them were developed for cardiology clinical trials. An example of a method that was developed by, in the early days of clinical trials, was group sequential methodology, which was developed by members of my group before it was my group. So that's one of the real contributions, because the interim looks at the data are very important for establishing whether a treatment is already efficacious or if in fact it's completely non-efficacious. And added to that, of late, has been futility analysis to make sure, to assure that nothing is really ever going to happen. So there's a little bit of a problem with that. If you decide a trial which is still accruing patients is not going to yield positive results, that of course would be less credible than if you had all the patients in the trial. So that's a bit of an ethical dilemma. We have methods now to change trials mid-course, either increasing the sample size because we estimated an effect size that was inappropriate, too small, or some of the assumptions we made in our sample size calculation were wrong, and we need to increase the sample size because of that. So that's one change. There are a lot of methods for different kinds of adaptations, and some of them are Bayesian. So I think if you want to know what the future holds, I think we'll be doing more adaptive trials, and I think we'll be doing some Bayesian trials, although they haven't caught on in cardiology. And both of these things are used in cancer. Cancer seems to be ahead of us in a certain sense. So.
Well, you know, as we've moved forward in cardiology, we're measuring all these different intermediate variables in clinical trials, and then I guess we give the data to the statistician, and we're like, well, tell us what this means.
Please come and see the statistician first.
Yes.
So at least we should know your study is adequately powered to answer the question that you're really interested in answering.
So do you think that I know about Born-Ferroni connections and things like that, but do you think that moving forward from that, I have some background in genetic studies where you have thousands and thousands of data points. So do you think that how are we going to be handling those things as far as like trying to actually make some sense of what we find?
Yeah, there are also a lot of new statistical methods for genetic studies. We could do somewhat better than Born-Ferroni, but I haven't been working so much in that area. But there are a lot of people developing methodology. In fact, we find a shortage of clinical trialists in the biostatistics field because everybody's doing genetics. It's a new hot topic.
So my last question for you would be that you have a different perspective on these trials because you kind of take them apart as far as the stats is concerned. So do you like children? Do you have any like favorite clinical trials in cardiology over the last like that were just done so nicely and like answered the question really well?
That's hard. Now, since I work for NHLBI, I have to,
you can't do an industry trial. Yeah.
It should be an NHLBI sponsored trial. Well, I guess I'll answer in a funny way. I haven't seen very many positive clinical trials, and actually, the last trial I saw that was positive in my own experience was Peace, which was published in the 90s. Until very recently, I did work on the Freedom trial, although I'm not a co-author. So that's a positive trial of late. So that's always very exciting. Whether they're my favorites, I don't think I have a favorite. I love them all.
Well, thank you so much for sitting with us and talking about it. But it was really a pleasure to talk to you. And hopefully, you're saying that there's not that many people doing clinical trials, so maybe there'll be a job for me when I'm done.
I think there will.
Thank you.
Welcome back to the Duke Cardiology Fellows blog. So for the first time in December, we're going international. We're going to Paris, France, at the invitation of Dr. Fayez Zanad, who joins us today. He's a professor of cardiology at the Nancy Clinical Investigation Center, and we're here in Orlando catching up before Cvct Forum, which is in December, which we're both very excited about. What are you looking forward to at Cvct?
Well, Cvct is a special kind of meeting. It's totally focused on clinical trials. This is what is the CT and the Cvct stands for, cardiovascular clinical trials. So it's mainly a meeting which is assembling all stakeholders in clinical trials, not only investigators, but also lots of industry, research and development people, and regulators. We do have EMEA and FDA people on board. We have NIH and NHLBI people as well. So all stakeholders, and it comes from doctors, cardiologists, but also statisticians, methodologists, and mainly discussing not only the big important trials, which are many of them have been presented actually here at the American Heart Association, but mostly about the interpretation, how we got there, what are the future trials, and also approvability and discussion on methodological, statistical issues. So it's really clinical trial science applied to cardiovascular area, and not much of promotional or highlighting what is hot into antithrombotic treatment, but what are the challenges with antithrombotic trials, for example, and childhood diabetes and all these cardiovascular trials.
To your point, one of the things I'm really looking forward to the CVCT is the small work group sessions where we'll have consensus-based documents that come out of that. One of the ones that I'll be working on is on antithrombotics and heart failure. What should I be looking forward to in terms of that work group?
Absolutely, and this is what is very special in this meeting. It's more about brainstorming, reaching some consensus rather than lecturing and didactic. So eventually some new ideas come out, and this is what we like, to capture them and to put them forward. We have got a WebEx, we have your blogs, and now we will also draft manuscripts. We have been successful so far into drafting position papers co-authored by these really important trialist people, these highly expertise, so that to set the stage, and these papers have been very contributed to really moving forward with the area of trials and methodology.
There's certainly a lot to look forward to, Dr. Zanad, at that meeting, so I hope you all stay in touch with us as we report from that meeting. Thanks again, Dr. Zanad.
Thank you.
Professor Bart Stills from the University of Lille provided an excellent summary of the HDL hypothesis in cardiovascular disease and the role of CETP inhibitors.
And one of the reasons we believe that HDL has this cardioprotective activity is due to its so-called antiatherogenic actions. And
he discussed the current CETP inhibitors in development.
And so, as John mentioned, there are now 3 different CETP inhibitors, or newer CETP inhibitors at different stages of clinical development, anacetrapib, dalcetrapib, and more recently we also got data on this new compound, evacetrapib.
He noted that they did not appear to have an adverse effect on blood pressure as seen in torcetrapib.
I'll show you this slide, which quite clearly shows that unlike torcetrapib, the newer CETP inhibitors are blood pressure neutral.
He summarized the talk by saying that in the case of HDL, higher might be better.
The feeling of the specialist is that the concept, which comes from epidemiology, that the higher the HDL cholesterol, the better, is progressively being changed towards a concept, the faster the turnover of the particles in reverse cholesterol transport, the better.
Next, Professor Chapman, director of the Dyslipidemia and Atherosclerosis Research Unit of the National Institute for Health and Medical Research, INSERM, spoke about apomimetic peptides.
An amphipathic peptide is a peptide with a helical conformation, an alpha helix, with a hydrophilic face with charged amino acid residues, and a hydrophobic or more lipophilic face, obviously with affinity for binding lipid.
He discussed the importance of these novel agents.
There are a number of advantages with these peptides: low production costs, infectious agents can be avoided, and endotoxin contamination particularly, which raises the cost of producing these recombinants.
He told the audience about why apolipoprotein A is so fascinating.
Carotid IMT, or the likelihood of angiographic coronary disease. And the efflux capacity was more strongly associated with those functional parameters than was HDL cholesterol.
He spoke about how the peptides can differ in their function.
And expect to obtain perhaps a large spectrum of functions, biological functions, of a single peptide.
And then he gave a summary of what is known at this time.
So I think I'd just simply like to summarize. None of the peptides studied at least to date, and certainly in the studies of Allen, has been equally effective in antiatherogenic functions. and each function appears to have its optimal set of structural features. And clearly then, this places us in the context where different functions of HDL may have different mechanisms and ultimately structural features.
Last, Professor Robert Rosenson, Director of Cardiometabolic Disorders at Mount Sinai Hospital in New York, spoke about a novel drug called RVX-208, a first-in-class small molecule that increases the production of ApoA1 protein, which in turn is used to make new HDL particles.
RVX-208 is a small compound that falls in the quinazoline family. It's got excellent, bioavailability, at least in green monkeys, of about 44%. Increasing concentrations of RVX-208 are associated with increased synthesis of ApoA1 protein and increase in the ApoA1 protein, as we can see here.
He showed the results of the phase 2 study in humans, the ASSERT study.
Here there were 300 patients that were randomized to a placebo or 3 different doses of RVX-208. In this study, we can see that the highest dose was associated with the greatest increase in HDL cholesterol, but we're talking about a concentration of only 8% on the quantity side. This was predominantly due to an increase in the large cholesterol-enriched HDL particles.
He concluded that several questions remain unanswered and larger studies will be needed.
The increases in ApoA1 and HDL cholesterol were modest, but this is a measure of quantity. The quantity doesn't necessarily impact in terms of the quality, because we saw that there was an increase in the pre-beta HDL that effectively caused efflux from macrophages via the 3 major pathways, ATP, A1, G1, and SRB1. However, we still have yet to learn whether these HDL particles maintain their antioxidant and anti-inflammatory properties. And maybe in the discussion, Dr. Chapman can tell us if he's doing any work with Reservologics on this compound, as that's his major area of research investigation. Liver function studies, again, are the major hindrance to the development of this compound. And again, looking at different vehicles may mitigate some of these potential risks. There are plans to evaluate this compound in patients with ACS, whether the ACS population is the right one for an ApoA1 inducer versus a population that's very high risk, let's say with diabetes, is one that I think is open to debate.
CVCT 2013 promises to be the most exciting meeting yet, with a star cast of attendees and an extraordinary agenda. We can assure you that the discussions will be pushing the envelope of developments in the field of cardiology. When they finally are translated to patient care, you will be able to say that you saw it first at CVCT.
Hi, I'm Ileana Pina from Montefiore in New York at Einstein College of Medicine, and we are here at the Cardiovascular Clinical Trialists Forum in Paris. This is a wonderful meeting where we really talk about clinical trials, practicality, new things on the horizon, and just have a very, very vigorous discussion. With me today is Chris O'Connor, who is head of the Heart Institute at Duke, and Carl Svedberg. You are now at?
Well, I've been since many years at the University of Gothenburg, but now I've changed to senior excellent professor from being only a professor.
So he's risen in the ranks. Yeah. We just finished a session on reducing heart rate and the importance of heart rate in heart failure, and heard some very interesting data from the SHIFT trial, which used ivabradine, which is a drug that is not available in the U.S., with some very positive results. So Carl, I want to go to you first, and if you could just summarize for our audience the results of the SHIFT trial.
Well, in SHIFT, the use of ivabradine resulted in a decrease of heart rate above placebo of 10 beats per minute, and that was associated with a significant reduction in the primary composite endpoint of cardiovascular death and heart failure hospitalization, and which was the driving effect in that outcome, which was reduced by 26%. So it was associated with very positive effects and very few side effects. So it was very well tolerated.
And on top of beta blockers.
Yes, that's important. So these patients, 90% of them already had a beta blocker, and everyone was had been considered for a beta blocker and not considered tolerating a beta blocker, those 10% who weren't on a beta blocker. And that's important, of course.
So Chris, part of the conversation was about the tolerability of beta blockers and how in practice doses that we know have worked in the trials are still not being used. Dr. Tavasi made a mention that all the heart rates are in the 70s, and I know that's what our heart rate was in HF Action. Can you comment on that as to why aren't doses better in practice?
Well, Ileana, first of all, this was a fantastic session.
It was great.
Congratulations, Carl, on your new title.
Yes.
This is a great honor. I don't think people fully appreciate this, but it's another 20 years of research for Dr. Swedberg in this position. But we still see, even in HF Action, although pretty well treated, mean dose of 38 milligrams of Carvedilol equivalents, there's still opportunity for titration. And I think, as Carl pointed out in the session, as he advocates, how do we position this therapy? We've got to make sure the beta blocker doses are A, on board, and B, at good evidence-based doses.
And ACE inhibitors.
But one of the analyses from HF Action that you've been involved with, Dr. Fuzat is leading it, has showed that once you get to evidence-based doses of about 50 milligrams of Carvedilol equivalents per day, there doesn't seem to be an increase in advantage at these super high doses. And so I see that as a very important space for a drug like ivabradine. The other space, Carl, I think I'd like you to comment on, 25th anniversary of Consensus, probably the highest mortality trial ever done, a mortality trial done in 250 patients. Ivabradine has an appeal because it doesn't drop heart rate, blood pressure. And so maybe the sicker patients, you could use this drug to get on because they're going to have higher heart rates to begin with.
You can titrate.
Thoughts on class 4?
It is quite possible that would be a target group, but we don't have the data. The results of the trial, the clinical implication are, I would say, that be concerned about heart rate. If heart rate is above 70, try to do something.
Exactly.
In Europe, we have ivabradine, not for heart failure yet, but so my message is that, but at least you should try to increase the dose of beta blocker. That is not being considered very much. And we saw in the trial a huge geographic variation between the 35 countries involved about the background dose, which is remarkable. But all these patients were particularly questioned, or the investigator were particularly questioned about the background dose of beta blockers, and they had to sign off reasons, etc., for why the patients were on a particular dose. So we were very meticulous about this.
And we know that in the U.S., the majority of heart failure patients are not seen by the heart failure teams, perhaps not even by cardiologists. They're seen by primary care. And so we really have targeted efforts to educate the primary care on how to up-titrate the beta blockers and do it safely. And I see the trend in the U.S. as we're moving more and more to additional physician extenders, pharmacists, as we see Dr. Fuzat, who's so involved with the HF Action trial, nurses, NPs, PAs, to actually go ahead and protocolize the increase in beta blocker without all the added questions that we as physicians say, well, what if this happens and what if that happens? You go through a protocol that's well vetted. And where I am now, we are starting a pharmacist-run clinic to up-titrate doses of drugs in the patients who have been admitted with heart failure within a 7 to 10 day block of time. So I think that that's an important message to the audience here, is that we need to think of that team approach to heart failure, and maybe that team approach will get the doses where we want them. Chris, any more comments on?
Well, Carl, one of the things that came up was the sudden death question and arrhythmias in general. The SHIFT trial really wasn't powered to detect a difference in sudden death. What are your thoughts on arrhythmias in the SHIFT trial?
Well, it wasn't powered to show an effect on death at all. Now, there was a reduction in deaths, and it was significant for deaths from heart failure. There was no effect on deaths due to sudden death, and we think, obviously we don't know, but our explanation is that because of the high background dose of beta blockers, that type of effect was already taken care of by the beta blocker therapy, which reduced sudden death.
Did you have ICDs in that trial?
Very few, and that was reflecting the practice in Europe and Eastern Europe for the type of patients included in the trial.
I think in the U.S. we would see a higher percentage.
And by design we had very few CRTs because patients could be paced too much, of course, because then we couldn't reduce heart rate.
Right.
So the devices were fairly uncommon, only about 3%.
Right.
Kim Fox gave a beautiful overview of the history of heart rate and even showed some data showing that increased heart rate is harmful with these new weight loss stimulants. Dr. Pena drinks a lot of caffeine, which we know increases heart rate. Is that risky, Carl? Does caffeine increase the risk of?
Save your beats, and if the coffee. Actually, I was in the DSMB of the SCOUT trial with the sibutramine, and we did observe early on that the effect on the harmful effect on MI, which and at the same time as there was this increase in heart rate. So increasing heart rate is not good. Decreasing is probably good for everyone, and in particular in patients who have heart failure.
So I'll make a plug for exercise. Exercising increases your heart rate while you're exercising, but at the end it improves your vagal tone, which is thought to be one of the benefits of exercising, and I don't drink that much caffeine.
Well, the take-home message, Carl, sounds like a heart rate of 70 maybe where clinicians should push below, beyond.
We should, the target, based on our experience from the trial, is 60 beats per minute. So if you can, but for sure, be concerned if it's above 70.
And tell you that you still have room to go. Take a look at all the drugs and think about up-titration even when you think that you've reached the maximum dose. And we're learning this from our BMP-guided trials. So I'm going to wrap it up here, and I want to thank our audience. This has been a lot of fun. I want to thank Chris and Carl for joining us today, and our brilliant cardiology fellows for doing the filming. Have a great day.
Welcome back to the blog. I am pleased to be joined today by Dr. Bertram Pitt, who is Professor of Medicine Emeritus at the University of Michigan School of Medicine, who has really been one of the pioneers in the field of aldosterone antagonism in heart failure. I'll be joining him later in December at the Cvct Forum in Paris, France, where we'll be certainly talking a lot about aldosterone antagonism in heart failure. From the ROUSE trial to now, and then looking beyond, what do you see as far as aldosterone antagonists and their implications for use in clinical care?
Well, I think we've already shown that in systolic heart failure, both severe and mild, that it's very effective in reducing total mortality and hospitalizations on top of everything. And what we're waiting for is the results of TopCat in heart failure and preserved systolic function. But regardless of that, I think we haven't scratched the surface of aldosterone blockade. We don't know the answers in acute decompensated heart failure. We don't know the answers in prevention of heart failure. And then beyond heart failure, there is clear indications that aldosterone blockade will be useful in patients with infarction independent of heart failure, probably useful in atherosclerosis, the metabolic syndrome, prevention and treatment of renal disease, and several other areas. So this is a wide open area for the future. We've been limited, unfortunately, by the fact that spironolactone has been generic for 40, 50 years. Eplerenone is now generic in the U.S. So getting support for future studies is difficult. We are supported by the NIH in the TopCat trial, but these days large-scale trials and the money is very difficult. So our hope in the future is that there are a number of non-steroidal mineral corticoreceptor blockers that are now being tested with very long patents, and if any one of those agents is shown to have an advantage, and mostly people are looking at a safety advantage. If they have a safety advantage and they have a long patent, then some of these things we talked about will be explored. Without that, I'm afraid it's going to be a very difficult time. So we're very much dependent upon commercial development and patent life, given the current climate of limited economic resources. But if one of those things comes through, which we're hopeful they will, then I think the future is very, very bright.
It's my opinion, I'm sure I'm not alone in this, that it's absolutely critical that we focus on expanding the investigation of aldosterone antagonists, because it seems like we really only scratched the tip of the iceberg in terms of its benefits.
Well, that's what I'm just trying to say, is that there are just so many places to go. I think the next decade will be a tremendous opportunity, both from clinical investigation as well as from basic science, because we're learning every day. I mean, every day we're learning something new about blocking the mineral corticoreceptor, effects on fibrosis and what's activating it and how to do it. But the clinical opportunities and clinical research opportunities are just fantastic.
Well, this is certainly a ripe area of discussion. I think you set the stage very well for the Cvct Forum in December, so I look forward to carrying those conversations on then.
See you then. Thank you.
Great. Thank you very much.